In my experiment, I utilized Artemis Salina as a model to examine the effect of acidification on a marine ecosystem. Now, just as some brief background information, climate change is brought about by the burning of fossil fuels, which creates carbon dioxide buildup in the atmosphere. And in 2019, this buildup reached a rate of 10 billion tons of CO2 per year. And in this graph from the National Oceanic and Atmospheric Administration, there was a 32% increase in the past 60 years of the atmospheric carbon dioxide levels, growing from approximately 320 parts per million to 419 parts per million. And researchers and scientists alike have agreed upon the idea that 350 parts per million seems to be a common threshold for the globe. However, the globe surpassed that nearly 30 years ago. And some of the CO2 gets dissolved in oceans, which leads to the idea of ocean acidification. Now, this is brought about by the dissolution of carbon dioxide, which forms carbonic acid and later dissociates into hydrogen and bicarbonate ions. And this buildup of hydrogen ions leads to acidification and the decrease in pH due to an elevated level of H+ ions. And this lack of carbonate ions, which are necessary for growth and development in marine organisms, reduces the structural integrity of many marine organisms as they rely on it for necessary growth and development. Now, I used algae in my experiment to modulate CO2 concentrations because they're photosynthetic organisms that utilize 6 moles of carbon dioxide and 6 moles of water in order to produce 1 mole of glucose and 6 moles of oxygen. And my test organism was brine shrimp because they are heavily dependent on carbonates, specifically to strengthen their exoskeletons and skeletal-muscular system. And also they are primary consumers and very low on the food web. So a decline in this population would have a detrimental ecological impact on the marine food ecosystem. And this raises the question of how can Artemis Salina be used as a model to see the effect of emulated acidification on a marine ecosystem. Just as some background research, Taylor et al. 2015 investigated the impact on ocean acidification on shrimp and found that in a given time period, the exoskeleton growth substantially minimized and the mineral activity substantially decreased in the shrimp, indicating that a decreased pH level had an impact on the exoskeleton growth of shrimp. And Ho Goldberg et al. 2007 found specifically that a decrease in the pH had a substantial decrease in the population size of corals and other marine life because of a lack of available carbonate ions. So the purpose of my experiment would be to understand how a lower pH and continued CO2 buildup will harm marine organisms for future reference. For my methods, I utilized 2 external fermentation devices that supplied 2 tanks with an external supply of CO2, and this was created through a 14% yeast solution being mixed with a 60% glucose solution to create 2 contained respiration reactions. And as you can see on the left, these respiration reactions took place in plastic containers and passed through a system of filtration and tubing in order to ensure that only CO2 is being dissolved in the tanks on the right. I used 3 tanks in my experiment, a control, a negative control, and experimental. All were supplied with 2 liters of salt water at 1.028 specific gravity, and additionally Artemis Salina eggs. However, in the control, there was Agar or algae. The negative control tank contained CO2, and the experimental contained Agar and CO2. In my methods, I utilized an O2 probe and a pH probe in order to measure fluctuations in the dissolved oxygen and pH probe over the course of the trial. And these measurements were taken at the beginning and end of each trial. And and additionally, the population of living and deceased Artemis Salina was taken each day. For my results, specifically honing in on trial 3, there was a substantial decrease in the pH in the negative control tank from 6.38 to 5.5, and simultaneously a decrease in the population from 39 to 2. However, in the experimental tank, it only went from 6.25 to 6 in the pH, indicating that the algae may have played a role in combating the elevated CO2 levels, and there was not as much of a decline in the population from 39 to 25. Additionally, the highest rate of survivability was found in the control tank as the pH actually increased and the population went from 31 to 25. In conclusion, brine shrimp had the lowest survivability under carbon dioxide, and it was greatest when exposed to just algae. However, the brine shrimp survived at a greater rate when exposed to carbon dioxide and algae when just exposed to carbon dioxide compared to just carbon dioxide. This proposes the idea that algae may have served as a combatant, and however, marine populations will experience great loss if the CO2 dissolution continues in the future. However, my experiment contained many variables, so I cannot isolate CO2 or pH as being the catalyst for death in this experiment, and the sample size was too small in order to be deemed statistically significant. This is my literature cited. Thank you.